Ya estamos. Bueno, esta presentación va a ser en inglés, nada más como aviso a todos los participantes. Welcome. This morning we have Dr. Juan Vivero-Escoto. He's an associate professor of the chemistry department at the University of North Carolina, Charlotte. He received his bachelor in science and master in science in chemical engineering from the National Polytechnic Institute of Mexico and has a PhD in chemistry from Iowa State University. He was a postdoctoral fellow through the Carolina Postdoctoral Program for Faculty Diversity at University of North Carolina, Chapel Hill. Dr. Vivero-Escoto has numerous professional honors, including the Ralph E. Power Jr. Faculty Enhancement Award from Oak Ridge Associate Universities, the Wells Fargo Faculty Excellence Award, the Class Junior Faculty Development Award from UNCC, the Outstanding Faculty Award, the National Science Foundation Catalyst Fellow, the National Science Foundation AGAP-NC Fellow, and the AAM Innovation Award. His research focused on the development of silica-based nanoparticles for biomedical applications. Dr. Vivero-Escoto's group has published over 30 peer-reviewed papers, book chapters, and editorials. And we are lucky to have Professor Vivero-Escoto today for this webinar that is part of the Latin American Network of Nanoscience and Nanotechnology webinars, and this is the last one for this year. And today he's talking about multifunctional silica-based nanoparticles for cancer treatment. Thank you very much, Juan, and please start your presentation.
Okay, thank you, Dr. Mendez Rojas for the kind invitation, and Dr. Irene for arranging this, for allowing me the opportunity to present the research that my group has been developing on the synthesis and application of multifunctional hybrid silica-based nanoparticles for cancer treatment. This is the outline of my presentation. I'm gonna
Juan, we cannot see your presentation.
Okay. Let me. Sorry for that. How do I share it? Okay, let me try again. So I'm sharing again. Let me know if you can see it.
Yes, we can see it now.
Okay. And I making active. You still can see it?
Yes.
Yes.
Yeah. Okay. Yeah. Sorry for that. Apologize for that. I thought I shared before. Yeah. So this is the outline. I'm gonna focus my presentation in two main platforms that we have developed in my group. Sorry, I just want to put the laser pointer. One platform deals with the development of polycell cis-coxane nanoparticles of PCLQ, as we call in the lab, and also mesoporosilica nanoparticles, MSNs or MSNPs. I'm gonna give you two examples to illustrate what is the approach that we use in my group to develop these platforms and to apply these platforms mainly for cancer treatment. I'm highlighting here the two types of cancers that we are focusing for these platforms. I won't go too much in detail for this, the biology of this type of cancer. I just want to highlight here the main problems or the general problems that these cancers are bringing for their treatment, which is triple negative breast cancer, as indicated here, and pancreatic ductal adenocarcinoma, which both, mainly PDAC in particular, has a poor diagnosis. Both of them, there are very few alternatives for treatments, and usually those treatments are result in a very poor prognosis. Both of them will develop drug resistance for the different or the standard type of care for the treatment of these type of cancers, and also there is metastasis. So there is a clinical need to develop new strategies to treat this cancer. And again, most of or all the presentation will be focused more in the material science and chemistry that we use for develop these nanoparticles. Okay. So just introducing again these two platforms, which is mesoporous silica. In the lab, we develop different type of mesoporous silica. Most of the research that I present today will be using what I will call the typical type of mesoporous silica, spherical shape with a size of 40 to 50 nanometers in diameter, pore size of 2.5 nanometer, surface area around 800, 1,000 square meter per gram, and also polycell cis-coxane nanoparticles, which also are spherical in shape. The first part of the presentation, I will focus in the polycell cis-coxane nanoparticles. So why we like polycell cis-coxane nanoparticles? So we believe, and well, of course, not only our group, other groups in the field believe that these are platform that has a very high degree of customization in the sense of the chemistry and the functionalization of these platforms. For instance, we can develop multifunctionals and multimodal systems through integrating very quite different components to the platform, to the network. We can incorporate drugs, anti-cancer drugs, photosensitizer into the network, as indicated here in the cartoon. We can use stimuli-responsive linkers to develop that network. We can include imaging agents or other type of therapeutics like DNA, siRNA, and we further functionalize these nanoparticles with polymers or targeting agents, depending of the application of the platform. These platforms usually have a high loading capacity for the different targeting, the different active components. They are biodegradable or can be engineered to make it biodegradable and biocompatible. In the group, we have used this platform, PCLQ, for different applications in terms of the cancer treatment, chemotherapy, gene therapy, photodynamic therapy, immune therapy, and one of the most recent, or at least one example that I'm going to show you from my lab is combined therapy. Okay, so this project, which actually was published last year in ACS Applied Materials and Interfaces, combine three different types of active components in the same platform, in the same PCLQ nanoparticle. We use a curcumin as an example of an anti-cancer agent. We use a porphyrin, protoporphyrin 9, or a derivative of protoporphyrin 9 as a photosensitizer. And we also designed the system to carry RNA for gene therapy. So I'm going to show you quickly what is the approach from the point of view of the synthesis of the main component of the network. In this case, what we design is an active component that, or in our particular case, is going to be an photosensitizer, and I just will show the photosensitizer that we use, which is protoporphyrin 9. That photosensitizer is chemically functionalized in such a way that we add three main components. One, an extra functional group, which in this particular case, we added a carboxylic acid, or I guess 2-carboxylic acid to the molecule for further functionalization of the nanoparticle, and stimuli-responsive linker, which in this case, we have a disulfide bond and tritoxysilane or trialkoxysilane. In this particular case, is tritoxysilane, which is the functional group that we use, or the group that we use for polymerization of the monomer, in this case, a monomer, to make the nanoparticle. And at the end, using either reverse microemulsion or microemulsion approach, we can get nanoparticles through this approach and using this as I mentioned before, as a monomer, and that will be the main component of the system. Something worthy to mention in terms of the protoporphyrin 9, well, it's clinically approved and is one of the most common photosensitizers for photodynamic therapy. So that was another reason that we picked this porphyrin. So as I was mentioning, using the protoporphyrin silane derivative as a monomer, we synthesize the nanoparticles using reverse microemulsion or microemulsion. In this particular case, we use microemulsion. I will show some characterization of these nanoparticles in the next slide, but well, we get a spherical nanoparticle, and we the version that contains the curcumin, we co-condensate both the protoporphyrin 9 and the curcumin in the same synthetic approach in this microemulsion approach. The nanoparticles was further functionalized with polyethylenimine through electrostatic interaction. As I mentioned before, one of the functional groups in the system is carboxylic acid, which under physiological conditions are deprotonated, making carboxylase, which gives a negative charge to the overall surface of the nanoparticle. So we took advantage of that negative charge to interact electrostatically with polyethylenimine to cover the surface of the nanoparticle, and that nanoparticles was further functionalized with polyethylene glycol. And polyethylene glycol is mainly to increase for in vivo applications. I won't show data, at least for this system in vivo application, but in general, for in vivo application to enhance the blood circulation time and also the biocompatibility of the system. And of course, one of the ideas to use this the positive charge coming from the PI was to have an electrostatic interaction with the siRNA to use the platform as a carrier for siRNA. So in this case, we have the three components, as I mentioned before: the photosensitizer, the anti-cancer drug, and the RNA that is integrated in the same nanoparticle. Characterization, well, TM, SEM showed that these nanoparticles, as I mentioned before, are spherical in shape, around 40 plus minus 5 nanometers in diameter. Dynamic light scattering shows some aggregation, most likely due to the presence of these hydrophobic porphyrins. The content of porphyrins in the nanoparticles is around 20–25% in weight, and in the case of the curcumin is between 5 to 7% in weight. So it's a high content of the active components, which again, that's reflected also in the physical properties of this system in terms of the aggregation. But the aggregation is not that critical, in the sense we are not talking about nanoparticles that are aggregating in the micro size regime. So it's still aggregation that we can handle, at least for in vitro and in vivo applications. The zeta potential show some of the features that I mentioned before in terms of the surface property of these systems. At the beginning of the fabrication of these nanoparticles, the surface is negatively charged. Once we interact or we modify the surface with PI, is changed to positive charge, and PEG just passivate a little bit further that charge in the nanoparticle. We tested these nanoparticles in triple negative breast cancer, MDA MB231 cells, and using the dose response approach, or the dose response curve for the phototoxicity of the systems under these irradiation conditions. And we determined, using these dose response curves, we determined the IC50. So the main take-home measures that we got from the IC50 for the different nanoparticles in comparison with the parent photosensitizer and the parent anti-cancer drug, which is curcumin, is that the nanoparticles has a similar phototherapeutic response, or IC50 in this case, as a way to measure that phototherapeutic response as the protoporphyrin 9. But there is a synergy when we incorporate the curcumin inside of the nanoparticle, meaning the IC50 decrease compared with that, compared with the protoporphyrin 9. That's an indication of this additive effect. And also what is important is the toxicity of the actual protoporphyrin 9 under dark conditions is reduced by incorporating this protoporphyrin 9 in the nanoparticle. Of course, one of the principles of photosensitizer is that shouldn't be toxic under dark conditions, only when they are irradiated, but as any chemical, at certain concentration, they start to produce some toxicity. So also what we found, as I mentioned, is that by incorporating the protoporphyrin 9 in the nanoparticles, the toxicity in dark is reduced for the parent porphyrin. Next, we tested the properties of these nanoparticles for delivering siRNA. In particular, as a proof of concept, we use cells, triple-negative breast cancer fluorescence protein. So we tested the ability of the system to silence that green fluorescent protein. So using flow cytometry and or nanoparticles at different concentrations, we determined, or we saw that there is a dose response in terms of the silencing of that GFP. And in particular, the nanoparticle that is functionalized with PI has a higher response or higher silencing of the of those cells. We have fluorescence microscopy that show visually that silencing of the green fluorescent protein in this type of cells. And it's an indication that our nanoparticle not only can produce phototoxic effect and chemotherapeutic effect, as indicated in the previous slide with the protoporphyrin 9 and the curcumin, but also can efficiently deliver the the siRNA to silencing. As I said, in this particular case, as a proof of concept is the silencing of green fluorescent protein. Okay, so moving to the next example for this platform. In this particular case, we are showing the capability of these polycell-cis-CoV nanoparticles with two goals, or at least the goals that I will show for this presentation. One is to increase the degradation of these nanoparticles, and also to demonstrate that there is a phototherapeutic effect in an animal model. What is important to increase the degradation? Some of you might know one of the main criticisms for inorganic nanoparticles, in particular case for silica-based nanoparticles, is that the degradability of these nanoparticles is slow compared with nanoparticles that are already tested in the market, like liposomes or polymeric nanoparticles. So it's important to develop strategies to enhance that degradability for this type of silica nanoparticles. So what we did in this case is we are using the same monomer as I described it before. Just remind you, the disulfide bond is an important player for the function of these nanoparticles in terms of the degradability. We changed a little bit the strategy for synthesize the nanoparticles. In this case, we use reverse microemulsion method, and the functionalization with PEG was directly taking advantage of the carboxylic acid and making an amide bond to functionalize the nanoparticle with polyethylene glycol. So in solution, we tested the degradability of these nanoparticles using a reducing agent. We are showing here the DLS of the nanoparticles before adding the reducing agent. We can see here the hydrodynamic diameter of the nanoparticle. And after that, we add the reducing agent, which is the solid line. And we can see after a certain time the hydrodynamic diameter starts to shift to smaller sizes as an indication that the nanoparticle is starting to degrade, to get some small pieces, let's put it that way, of the nanoparticle. Why is important this? Because as some of you might know, well, one of the ways to increase the degradability of the excretion of the nanoparticles in the body is by targeting the renal pathway, which usually is very fast in terms of the excretion of components, but there is a threshold there in terms of size, which usually below 8 nanometers, nanoparticles below 8 nanometers, or molecules. Of course, molecules are much smaller than that, but anyway, nanoparticles below 8 nanometers can be excreted through renal excretion. So that was the logic behind developing this system that degrades, that start to produce smaller pieces of that nanoparticle that can be excreted by that renal excretion pathway. We also evaluate the release of the protoporphyrin IX as an indication that the system is also, in this case, a redox responsive system for the release of protoporphyrin. We move forward to testing this system in vivo. This is immune competent mice or mouse, where we tested the degradability of our nanoparticle. For that, we incorporate a near-infrared dye in the nanoparticle, and we can see here we define two different regions: the abdominal region of the mouse, and the region where most likely the bladder is located, and we follow the luminescence of the nanoparticles, or in this case, the near-infrared dye associated to the nanoparticles at different times after intravenous injection of the nanoparticles. We can see that in the abdominal region, there is an increase in the accumulation of the nanoparticles, followed by around increase maximum at 4 hours, followed by a decrease after a certain time. And in the case of the region that is associated to the bladder, we can see that there is an increase in the accumulation of that luminescence as an indication that possibly, as I mentioned before, these nanoparticles are being excreted to renal excretion pathway. After 10 days of administration, the animals were sacrificed and the organs were collected, and we analyzed two main components of the nanoparticle, silicon, using ICP-MS, and protoporphyrin IX using a method reported in the literature. And we compare in this particular case for silicon, for the analysis of silicon, with mesoporous silica nanoparticle as a control system that we know that the degradability is not that fast as other nanoparticles, or I guess a system that will degrade that fast. So what I want to highlight here is when we compare the amount of silicon in those organs, in particular liver, kidneys, and lungs, we can see that mesoporous silica nanoparticles, there is still an accumulation in that organ, in liver, and also in lungs. And in comparison with our nanoparticles, these are three different concentrations of our nanoparticles. We see that the amount of silicon is similar to the control or negative control, which are animals that were not injecting with the nanoparticles, as an indication that most of all the nanoparticles has been efficiently excreted after 10 days. We also look at the amount of protoporphyrin IX accumulated in those organs, and again we can see that the amount of protoporphyrin IX is similar to negative control, again the animal that was not injected with this type of nanoparticles, again as a further confirmation of the fast excretion of these nanoparticles in the system. Finally, we tested the therapeutic efficacy of these nanoparticles in an orthotopic model of triple-negative breast cancer. The administration, the regime for the administration and treatment of these animals follow 5 injections of the nanoparticles, which were irradiated 3 times after the injection at different times. We measure the tumor volume of these animals, and we can clearly see that there is a decrease in the growth of the tumor for the animals that were treated with our nanoparticles and light, comparison with just the protoporphyrin IX and light, and also the nanoparticles that were not irradiated, and of course just the control that was not treated. This performance was confirmed by the tumor weight after the animals were sacrificed at the end point. Also the group that was treated with our nanoparticles and light showed the best performance in terms of therapeutic efficacy or phototherapeutic efficacy. We did an analysis of the apoptosis in the cells in the tumor region, which is very important using immunohistochemistry, and we can see that again the group that was treated with the nanoparticles in light showed this the highest population of cells that show apoptosis as an indication of efficient treatment for this tumor. We follow the toxicity and the biodistribution of these nanoparticles. There are not major toxicity, at least by the weight of the animals during the whole treatment for any of the groups. In the biodistribution, we saw some interesting results where we can see that the group that was treated with protoporphyrin IX, actually there is a high accumulation of protoporphyrin IX in lung and liver. It is reported, as I say, protoporphyrin IX is used in humans either as ALA or MALA or derivative of ALA or MALA that are the precursor of protoporphyrin IX, but it's reported that usually protoporphyrin IX accumulates in liver in humans, and there is some toxic effect. Even though we don't see that toxicity in these animals, as I mentioned, it's important was important for us to notice that there is that accumulation of the protoporphyrin IX in these organs, liver and lung, actually happens. And of course, the accumulation of nanoparticles in tumor was much higher for our nanoparticles than for our control experiment. So as conclusion of this first part of the presentation, well, I show you that PCLQ nanoparticles can be functionalized and modified to incorporate different active components. In this particular case, I show you that we can incorporate a photosensitizer for photodynamic therapy, an anti-cancer agent, in this case curcumin, for chemotherapy, and also can efficiently deliver siRNA for gene silencing. Also, the in vivo data shows that nanoparticles can be degraded, or the degradation of nanoparticles can be enhanced by engineering the chemistry of the network by adding that disulfide bond. And the system was still, I mean, even the degradation was improved, the system was still efficient from the point of view of the phototherapeutic application. Okay. So switching gears to the other platform that I will present today, mesoporous silica. Again, mesoporous silica, well, we see a lot of advantage on these nanoparticles. Is similar in terms of the functionalization of the system as I present for the PCLQ. We have the same advantage. In this particular case, well, mesoporous silica is the scaffold, and you can functionalize that scaffold with different active components. But also there is a high loading capacity for the system, and is biocompatible. And in the group, we have used this nanoparticle for cancer treatment, similar modalities that the ones that I presented for PCLQ. The first example that I want to show you to illustrate the work on a group with mesoporous silica deals with the development of nanoparticle that carry doxorubicin, and also carry novel nanoparticles that are called NAMs, or nucleic acid nanoparticles. So the idea behind this project was to take advantage, again, as I mentioned, of both platforms as delivery system, of course, MSN as a delivery system, and the NAMs as a nanoparticle to carry siRNA. And also what we wanted to test was the use of this platform for combination therapy, of course, chemotherapy and siRNA in particular, targeting cells that overexpress the anti-apoptotic gene BCL2. For some of you that might not be familiar with these nucleic acid nanoparticles, these nucleic acid nanoparticles are fabricated by my collaborator, Kirill Afonin, also here a colleague at UNC-Charlotte. And these are self-assembled nanoparticles, similar to DNA origami in the sense that you get nanoparticles containing, in that case, DNA. But in this case, the main components of the nanoparticles are siRNA, and they are used to transport siRNA with different type of siRNA in the same nanoparticles. That's the type of nanoparticles that we use for this project. And this project was recently published in ACS Applied Materials and Interfaces. Okay. So this is the description of the synthesis of the platform. Starting with the scaffold, mesoporosilica, we loaded doxorubicin to the scaffold. We use a similar approach as the one presented before for PCLQ, in the sense of taking advantage of the negatively charged of the mesoporosilica. We electrostatically bind polyethyleneamine, and followed by further functionalization with polyethylene glycol. The negative charge, the positive charge of PI will help us to have an electrostatic interaction with either just DNA, siRNA, or the NAMs that I presented before to have our final system or final platform. In this case, first, we optimize the M/P ratio. M/P is the amount of primary amines by the ratio of primary amines in the PI with phosphonates in the siRNA or DNA backbone using agarose gel. By using different ratios of the, in this case, we use DNA with the amount of PI, we determine that the optimal amount, in terms of the interaction between the siRNA or DNA with the platform was the ratio 10:1, 10 primary amines per 1 phosphonate. And thus the number that we use to calculate the loading of the different siRNA and NAMs during the whole experiment or the whole project. We also show using fluorescence, well, that the binding is stable under different conditions. There is not release, or there is not detach of that DNA or siRNA from the nanoparticle as a demonstration of the stability of the system. Also, something very important in the case of genetic material, nucleic acid material, is the stability under different enzymes, in this case nucleases. So our system showed that there is not degradation of the DNA in the presence of the nucleases, as an indication that our nanoparticle is protecting the DNA from any enzymatic degradation. We evaluated, in this particular case for these experiments, we just use DNA that was labeled with a specific fluorophore. So we use that DNA to test the internalization of the nanoparticles in triple-negative breast cancer cells. We can see that the nanoparticles are highly internalized using flow cytometry, and also confocal microscopy showed that the amount of internalization, of course, is concentration dependent, but both the nanoparticles and the siRNA that is being carried, because both were labeled, are internalized. Also important for us, by looking at the confocal images, we saw some release of the siRNA by the lack of overlap between the green of the nanoparticles and the red of the siRNA, which the overlap with yellow. So the absence of that yellow and the presence of red give us an indication that the siRNA is released from the nanoparticles in vitro inside the cells. We also determined that these nanoparticles are endocytosed and they follow an endolysosomal pathway, which go through early endosomes, late endosomes, lysosomes. Again, in lysosomes, we can see some release, escape of those nanoparticles, and release of siRNA based on the confocal imaging using the microscope. So now, once we determine as a proof of concept using this labeled DNA, when we move forward to using the NAMs, these nucleic acid nanoparticles, there are, my collaborator fabricate different type of nucleic acid nanoparticles, rings, cubes, and fibers. So we tested all these different type of nanoparticles with our mesoporous silica nanoparticle. This is the TM. The TM doesn't show that much in terms of the actual morphology of the NAMs on the surface, but we can see the difference in terms of the morphology in the surface, or the rough morphology in the surface of the nanoparticles that are naked, and in the presence of these NAMs after staining these samples for TM. Also, what important for us was to confirm that once the NAMs were loaded, electrostatically binded to our MSN, that interaction and the release of those NAMs from the MSN won't produce any degradation. So my collaborator was able to demonstrate by using heparin to competitive binding, or through competitive binding interaction, remove the NAMs from the nanoparticles, and showing that these NAMs nanoparticles that are released are still or were not degraded. They still have the same conformation and the same morphology and structure as expected. Okay. So now we use flow cytometry to test now the internalization of the whole system, or mesoporous silica nanoparticles and the NAMs, similar to the previous data where we just use DNA. All the material is internalized in the cells, and we don't see much difference between the different type of NAMs, the different morphologies. As a proof of concept for silencing, again, we use the MD231 that I described in the previous project using green fluorescent protein. In this case, we actually can see some difference in terms of efficacy, in terms of silencing efficacy. We can see that the fibers are actually more or has a higher silencing efficacy than the other two morphologies, which are the rings and the cubes. And this is just the double-stranded siRNA as a control for comparison. Okay, so these are just fluorescent images that show the actual silencing of that green fluorescent protein. So using that information, we designed, or my collaborator designed, fibers. As I mentioned, fibers were the more efficient in terms of silencing of green fluorescent proteins. So we use fibers to actually develop NAMs nanoparticles that will silence BCL2. And we combined that with doxorubicin, which was the whole purpose of the project, have this combination therapy. And we tested in two different type of cancer, melanoma and triple negative breast cancer. What I'm showing here is the data for melanoma. So we tested the toxicity the cytotoxicity of this combined therapy, we can see that there is an additive performance of our nanoparticles compared with other groups. We also tested the apoptotic effect of these nanoparticles, and we can see again that there is an additive effect by combining both the doxorubicin and the NAMT in our nanoparticle nanoplatform. And the last piece of evidence that we got for this experiment was the reduction of the expression, in this case, the BCL2 mRNA expression as measured by RT-PCR, which we see an increase in the reduction by using our combined therapy or combined platform. Okay, so that's using mesoporous silica for delivering NAMs and delivering doxorubicins. So let me move forward to the next project, which will be the last project that I will present today. So in this particular case, we wanted to develop, using mesoporous silica, a system where we can release two different types of chemotherapeutic drugs, which are actually very relevant from the point of view that gemcitabine, which this is gemcitabine, and cisplatin are drugs that are standard of care for different types of cancer. In particular, gemcitabine is standard of care for PDAC, for pancreatic cancer, and there are some encouraging results in clinical trials by the combination of gemcitabine and some platinum derivatives for the treatment of this disease. So we wanted to use our platform to deliver both drugs at the same time. So this is the rationale behind the design of the platform. We wanted not only to physically load these drugs, we wanted to have these drugs chemically conjugated to the nanoparticles using a stimuli-responsive linker. We also wanted to control the ratiometric loading and release, meaning the ratio between both drugs in the nanoparticle that is loaded and when they is released, to control that ratio. We also have a hypothesis that by incorporating the cisplatin inside of the mesoporous silica, or the pores of the mesoporous silica, and the gemcitabine outside, there will be a difference in the time of release of this drug. So as you will see with the data that I will show you, gemcitabine is released faster in higher amounts at the beginning, and cisplatin starts to have a slow release and after that reach a plateau. But that was important for us because we found in the literature that one of the advantage of combining gemcitabine and cisplatin is that gemcitabine seems to be that overcome some of the issues with the DNA repair mechanism, which I will talk a little bit more later for this project, but which are associated or highly associated for the resistance to cisplatin treatment. So by you taking advantage of the morphology of the MSNs, by having these two distinctive interior, or what we call interior surface area and exterior area, we wanted to control that release. And finally, we wanted to functionalize our nanoparticles with an antibody, which also comes from another collaborator in our university that targets specifically tumor-associated MUC1 in pancreatic cancer. So this is just a schematic representation of what I was mentioning before, in the sense that we wanted to control the release of gemcitabine, which are the blue dots there, followed by the release of cisplatin. That will increase the DNA damage and will avoid this mechanism of resistance, of DNA resistance for cisplatin. This is the synthesis of the platform. As I mentioned, well, we start with the scaffold, mesoporous silica. we use a cisplatin prodrug. Again, we wanted to have some chemical control for the release and the loading, and also very important, the toxic effect actually in vivo is very important to have a control for that. After adding the cisplatin prodrug, again, similar strategy as presented before, we coated with PI. In this particular case, we functionalized that PI with a linker that allows us to further functionalize the system, modify the system with the gemcitabine prodrug. Now, for this ratiometric ratio that I was mentioning in the previous slide, what we followed was a previous optimization in terms of what will be the amount, or what was the ratio that will give us the optimal therapeutic performance or outcome for the treatment of PDAC cells. So we use, in this case, the table is showing just the IC50 for the drugs that gemcitabine and cisplatin that were used to treat two different type of PDAC cells. We found that the optimal ratio in this case, using the combination index, was between 4 to 5.5 in terms of the ratio. So following this information, we engineer our nanoparticle to have a similar loading. And we ended with a nanoparticle that carries around 24% plus minus 2.6% in weight of gemcitabine and 5.1% plus minus 0.9 of cisplatin, which at the end give us a ratio of 5.3 plus minus 0.8, which is in the range of what we found for the cells. So characterization of the material, well, this is show characterization of the nanoparticles, what I will call naked nanoparticles. So this is just before functionalization, that, as I mentioned before, typical MSN, type 4 in terms of the nitrogen isotherms, high surface area, pore size of 2.3 nanometers, so nothing relevant from that point of view. Further functionalization, analysis of the nanoparticles, the material using DLS, showed that they are colloidally stable. PDIs below 0.3, so showing the colloidal stability. And this is the release profile of the drugs after adding a reducing agent. Because what is more important is what is happening at the first 5 to 6 hours. We can see, as I was explaining before, that was we were hoping to get based on the rationale for the modification of this system, that there is a very fast release of the gemcitabine, which is in the exterior surface of the nanoparticle. The cisplatin starts to release very, very slow, and which is the mechanism that we propose in this cartoon. Again, in the surface, the gemcitabine is released fast. There is more interaction with the reducing agent, and after that, cisplatin start to be released. But also what was important for us was to determine the ratio, which at the very beginning, meaning the first hour or 2 hours, we have a high ratio of the gemcitabine, which is indicative of this fast release of the gemcitabine. But after that, the ratio reached a plateau, which is in the same range of the optimal ratio that was shown before. So that was very important, that at the end we keep the same ratio, and that's during the whole time of the experiment. Okay, so with this system, we tested the cytotoxic effect using concentration response curves, where we determined the IC50 for this based on this information, and I show in the IC50 for the most important nanoparticles or controls that we have for this system. So we have the monotherapy, meaning nanoparticles that contain only cisplatin, gemcitabine. We have our system that carry both drugs, and also we have as a control an experiment where we actually combine both monotherapies at the same amount that the actual combined therapy in the same nanoparticle, just to further demonstrate that indeed carrying both drugs in the same nanoparticle actually makes a difference. So that's what we have this we call it physical mixture of the nanoparticles. And we tested these nanoparticles in 4 different cell lines, 3 human cell lines and a murine cell line, KCM. We showed that, or the data showed that their system that combine both drugs in the same nanoparticles is 2 to 3 times more efficient. So we wanted to further investigate why was more efficient this nanoparticle carrying both drugs compared with those controls. So what is happening with these 2 drugs, these 2 drugs, gemcitabine and cisplatin, actually target the DNA inside of the nucleus of the cells. Cisplatin forms adducts with the DNA, and gemcitabine gets incorporated in the DNA, in the backbone of the DNA, in the actual structure of the nucleic acid polymer. And that trigger, the first step that triggers is phosphorylation of the system or one of the proteins of the system. And after that is followed by the overexpression of different proteins. Of course, this is a more very complex process. So what I'm showing here is a very simplistic way to see this process. And I also highlighting these proteins because those are the ones that we follow in the experiments. So once the damage of the DNA starts by the phosphorylation, these 2 proteins will be increased as an indication that this damage is happening. And finally, there will be either a repair of the DNA or apoptosis of the cells, which it will bring the cells to death through apoptosis. And as I mentioned before, one of the important parts was a lot of the treatment with cisplatin usually produce DNA repair mechanisms that make this treatment not very efficient. So, and the idea of using gemcitabine in the same platform, or one of the ideas of using gemcitabine in the same platform, was to avoid that DNA repair mechanisms. So the preliminary experimental data that we have for this mechanism is that indeed the system that combine both drugs in the same nanoparticle enhance this phosphorylation as an indication that there is a higher DNA damage, which is also confirmed by the levels of proteins that we see after the treatment, in comparison, of course, with the control experiments. At the end, we see also a higher apoptotic effect of our material combining both drugs in the same nanoparticle, and even in comparison with, again, with this physical mixture that has these two drugs, or the cells were incubated with these 2 drugs, but the time of events when they are released seems to be that is very important to reach this therapeutic effect. So we still are investigating other proteins that are more specific for this DNA damage, that we hope that we can get more information about the actual mechanism that these drugs are using to avoid the repair of the damage through due to the cisplatin. So the next step for our system was to functionalize our nanoparticle with the antibody. As I mentioned, that was also one of our goals. For that, we use polyethylene glycol that was chemically attached to the surface of the MSN through the PEI. And we use an antibody, TAP004, that's the commercial name of the antibody that we use, which targets tumor-associated MUC1 for pancreatic cancer. Some characterization of this system, well, we'll see a colloidal, fairly good colloidal stability of the nanoparticles. There is an increase a little bit in the hydrodynamic size and also the PDI, but not something that worries us too much for that. For further in vivo applications, we stain the nanoparticles and we see the antibody on the surface of the nanoparticles, and we determine the amount of TAP in the surface, which that's 7.4 micrograms per milligram of MSN. Further characterization of the system for the targeting capacity, we use KCM cells. I guess I didn't mention about the features of these cells. These cells are obtained from a genetically engineered mouse model that was developed by our collaborator and overexpress this tumor-associated MUC1. That's one of the, I guess, the main features of these cells that overexpressed the antigen for the antibody. So we can clearly see by using flow cytometry that the nanoparticles that contain or were functionalized with the antibody clearly are internalized in higher amounts than the control, which is just the pegylated version in these KCM cells. As a further corroborate that indeed is the presence of this of this antigen, of tumor MUC1 antigen, that is the responsible for that higher internalization or targeting. We use another type of cells also coming from our collaborator, which are called KCKO cells, which in this case, these cells doesn't overexpress that tumor, that antigen, that tumor-associated MUC1 antigen. And we can see again the difference that when we're using KCM, there is a higher accumulation and compared with the KCKO cells as an indication that indeed this targeting is associated to the antigen. And finally, we also tested this targeting ability using cytotoxicity in KCM cells. We can see an enhance in the cytotoxicity associated to the TAP004 MSN system, and also in the HPAF2 cells, which are human cells, and also overexpress this tumor-associated MUC1 antigen. We can see that there is an increase, at least in 2 times in terms of deficiency, and also important, or we thought was important also to test a healthy cell line, and we can see that there is, at least at the concentration that we tested, which was 100 micrograms per mil maximum, there we didn't see any major cytotoxicity as an indication that the nanoparticles are also selecting cancer cells compared with these healthy cells. Okay, so as a conclusion, well, I show you that our group used mesoporous silica to functionalize and modify to carry 2 different types of therapy, chemotherapy, gene therapy, through dogs and these NAMs. We also take advantage of the systems, of the, I guess, beauties of the mesoporous silica, in the sense of that we can selectively localize the drugs in the interior or the exterior surface, and that can give us different spatio-temporal control for the release. We can also ratiometrically load and release these 2 important clinically relevant drugs. And of course, we can make a target-specific system using this TAP004. Okay, so, well, I would like to acknowledge quickly my group, the main people that did all the work for this research, and also the funding agencies for support of our research. And thank you for your time, your attention. I think I just kind of on time, or I guess I take a few minutes more of what was expected. Thank you, and I'm happy to answer any question if you have.
Thank you, Juan. It was a wonderful presentation. And I just found out that one of your graduate students is a former student of us, Paula Gomán Cortez. She's a chemist graduated from UDLA. So I didn't know that.
She is, yes.
Well, it's a great thing. Well, I don't know if anyone has a question. You can make the question in English or Spanish as you decide. We have time for a few questions. Por favor. Preguntas en español o en inglés, no hay problema. Pues, a lo mejor tengo que abrir los micrófonos. Ay, ¿cómo se hace eso?
Sí, no, ellos pueden directamente abrir su micrófono, Miguel.
Ah, ok, bueno.
A mí me gustaría preguntar sobre las vías de administración. ¿Cómo es que aplican esto? O sea, ¿cómo, si tiene que ser directamente al tumor o es por vía sanguínea, puede llegar al sitio de inyección?
La investigación que hacemos en el grupo lo hacemos a través de vía sanguínea. Es el principal método que estamos explorando. Ahorita digamos que porque es lo que se hace en el hospital, ¿no? Por lo menos para la quimioterapia. Estamos desarrollando materiales también para administración oral y en colaboración con un grupo en Brasil también uso para la administración a través de la dermis, de la piel. Pero toda el diseño que mostré en este seminario es pensando en que estos materiales son administrados a través de vía sanguínea.
Gracias.
Gracias.
Si alguien tiene pregunta, pues puede directamente levantar la mano o abrir su micrófono.
Creo que ya había. ¿Y Sofía?
Hola, buenas tardes.
Buenas tardes.
Mi pregunta es, ¿ya se tiene una idea o se han hecho estudios de cada cuánto se tendría que poner este tipo de quimioterapia intravenosa?
Bueno.
¿O cuántas dosis se necesitan?
Sí. Digamos, si la pregunta es relacionada con ya sistemas que tienen nanopartículas que son utilizados en clínicas o para quimioterapia, usualmente, o bueno, lo que entiendo, y ahí déjame decir que ignoro mucho porque, bueno, no soy médico, no estoy directamente tratando pacientes, pero por lo que entiendo es que empiezan con el mismo régimen que utilizarían para la droga. Por ejemplo, no mis materiales, porque bueno, estos materiales todavía no están en hospitales, pero te pongo, por ejemplo, Doxil, que es la versión liposomal de doxorubicina. Hasta donde sé, bueno, intentan o tienen el mismo régimen de tratamiento e intentan ver si se puede reducir. Ahí te puedo decir que una de las ventajas de la nanopartícula es que reduce los efectos tóxicos en el corazón y otros efectos tóxicos. Entonces ahí incluso hasta a lo mejor puedes incrementar la cantidad de la droga para tratar de ser el tratamiento más agresivo. Y aquí nuevamente te digo, no estoy en esa parte de la aplicación con pacientes. Entonces aquí estoy más suponiendo cómo lo pueden tratar ellos, pero creo que empiezan como los mismos regímenes que utilizan para la droga. Y de ahí van viendo ventajas y de ahí van modificando.
Gracias.
Gracias.
¿Alguna otra pregunta? Bueno, yo creo que por el tiempo, pues te agradece. Mildred, adelante, por favor.
Hola, muy bonita tu presentación.
Gracias.
Yo tengo una duda. Tú probaste estas nanopartículas solamente en un tipo celular, en lo que estoy entendiendo. ¿Tú crees que funcionen otros tipos de líneas celulares de cáncer?
Sí, bueno, la investigación que mostré lo evaluamos principalmente en dos tipos, que son el triple negative breast cancer y el cáncer de páncreas. Serían los dos diferentes tipos de cáncer que se evaluó. Incluso mostré algunos datos con melanoma. Creo que las nanopartículas sí podrían funcionar con otros tipos de cáncer. Ahí es ver más, bueno, si se piensa ir más allá me refiero a aplicar estas nanopartículas ya en hospitales. Ahí hay que ver, bueno, cuáles son las drogas que se están utilizando para esos cánceres, porque digo, si quieres lo que llamamos en inglés el translate from the bench to the bedside, es muy difícil que llegues con una nanopartícula a un doctor con una droga de gemcitabina, y si aparece cáncer, no sé, de colon, no se utiliza gemcitabina, se utiliza otra droga. Entonces ahí habría que modificar las nanopartículas con las drogas particulares que se utilizan para ese tipo de cáncer, pero en principio, digamos que esa es una de las ventajas, o por lo menos los que estamos trabajando en esta área, pensamos, una de las ventajas de las nanopartículas, que como sistemas de liberación de droga, bueno, se pueden implementar para diferentes aplicaciones sin modificar mucho el sistema, con la salvedad que ya mencioné. Bueno, la droga tiene que ser diferente y particular para el tipo de cáncer que se está tratando. Espero que haya dado tu pregunta.
Bueno,
sí,
pues
no sé, agarrar el micrófono. Gracias.
Gracias.
Pues muchas gracias, y pues ya estamos en tiempo. Juan, te agradecemos mucho tu presentación, que particularmente también me pareció muy interesante, y pues agradecemos a todos los participantes de este tercer webinar de la Red Latinoamericana de Nanociencia y Nanotecnología, y pues tu muy excelente exposición. Muchísimas gracias, Juan. Muchas gracias a todos. Gracias, y muy buenas tardes.
Gracias, un gusto conocerlo.
Gracias, igualmente.
Hasta luego.
Seguimos en contacto. Gracias.
Sí.